So good morning, afternoon, evening, and/or midnight to everyone tuning in for Biodiversity Genomics. I am Linelle Abueg, and I am a bioinformatician at the Vertebrate Genome Lab at Rockefeller University. So as the name of our lab might imply, we are one of the main production hubs for the Vertebrate Genomes Project, which I'll introduce you to here. As it says in the tin, aims to sequence all the vertebrates on Earth, and we are one of the consortium projects underneath like an umbrella of larger consortia, the Earth Biogenome Project, which aims at sequencing all eukaryotic biodiversity on Earth. We have a slightly smaller goal of only aiming at all 70,000 vertebrate species instead, and we are taking a stepwise phase-based approach towards this. So you'll see our schematic here. We are currently in phase 1, where we are getting a taxonomic representative from every order. And we aim to wrap this up probably at the end of this year, fingers crossed. And as part of this project, we also need to establish metrics for what counts as a high quality genome assembly. So we've previously established these minimum metrics of a contig N50 of at least 1 megabase pair, scaffold N50 of at least 10 megabase pair, and a quality value for base calling of at least 40. And I'm happy to report that now that we're working with PacBio high fidelity or HiFi data, we've been meeting these metrics routinely, and especially scoring higher than 40 for QV. So that leads us into our actual assembly pipeline, which takes advantage of this high accuracy PacBio HiFi data. So this is our assembly pipeline as an overall workflow schematic. We start off using HiFiasm here to make our contigs, and HiFiasm has a couple different modes that I'll touch on in a little bit, but either way, you'll use HiFiasm, you'll generate your contigs, and if necessary, you'll purge any duplicates, and then continue on to scaffolding steps. We usually do scaffolding with Bionano and with Hi-C data, but since our pipeline is implemented in a modular fashion, if you don't have any Bionano data, you can just, for instance, skip that workflow and just do Hi-C scaffolding. After scaffolding, the draft assembly is ready for decontamination, automated decontamination using a Kraken database, and also manual curation. And you can also run MitoHiFi to generate a mitogenome from the raw PacBio reads. So you'll note these little colors along the workflow. These stand for quality control metrics that our pipeline, as we have implemented in Galaxy, reports automatically, so the user doesn't have to do anything. It generates these QC metrics just like on the dot, and I'll just show you a couple quick examples of that. For instance, when we do an initial k-mer profiling to get an idea of the genome we're trying to assemble by using a genome scope plot. So these are generated as one of the first steps in the pipeline. And then after contigging, no matter what type of HiFiasm mode you're using, we run Mercury to generate, for instance, the spectrum assembly plot for, in this case, a homogametic specimen. This is with Hi-C phasing, by the way. And also other metrics such as Busco plots, as well as standard assembly summary statistics, which I've not included here because it's just a bunch of numbers. And then there's also QC for the scaffolding steps. We run pretext before and after scaffolding, so you can see the contact map and the effect of scaffolding. For instance, this is from a terrapin, and this pretext map is after Bionano scaffolding only, and this one is after Hi-C scaffolding of these Bionano scaffolds. So you'll see where Hi-C scaffolding has resolved some of these joins to create a more contiguous assembly as represented by this cleaner contact map. So I would just like to double back real quick to those different modes of HiFiasm. So HiFiasm can run in 3 different modes, depending on what supplemental data you have. The default one is run with only the HiFiasm reads of that individual. There's also a Hi-C phasing mode, which uses Hi-C reads from an individual. And there's a trio-based phasing mode, which uses maternal and paternal reads from the parents. We have switched to using this Hi-C integrated phasing as our default mode, and that's because we've been testing it with one of our lab's favorite animals, this little zebrafinch, and we've been seeing very good results with phasing on par with trio-based phasing. So we have trio data for this zebrafinch individual, and you'll see in this blob plot from Mercury this is the blob plot for that trio assembly. So the X and the Y axes stand for parental camers. So if a blob is closer to the X axis, it has more maternal camers, and Y axis more paternal camers, vice versa. And the color of the blob is corresponding to the assembly it comes from. So you'll see the trio base one is very neat, as one might expect. All the maternal, the red blobs are along the X axis, and all the blue blobs are along the Y axis. So this example of a blob plot is from primary alternate phasing, primary alternate assembly, so no phasing. It's a pseudohaplotype assembly, and this is post-purging. So the red dots, the red blobs, are going to be from your primary assembly. And you'll see, since there's a lot of these contigs that are in between the X and Y axis, indicating there's a mix of paternal and maternal camers within that contig, indicating haplotype switches. And on the contrary, in the Hi-C based phasing, it's a much more neat outcome compared to the primary alternate. It is, of course, not as clean as the trio-based one, but is certainly approaching that without trio-based data. So there is a mixture of contigs from the different parents in one assembly, but there's much fewer, at least, haplotype switches within the contig. So that's why we've been switching to using Hi-C phasing as our default for assemblies. And it also, because we have our projects always generating Hi-C data anyway, along with the HiFi, it just makes sense to be able to use this one as well, to be able to use this mode as well. And so you might be wondering how we actually use these workflows. We have implemented our pipeline on Galaxy, which is a public computational infrastructure that provides a robust and free analysis environment for investigators who might be limited by a lack of local computational resources or a lack of programmatic experience preventing them from taking on projects such as genome assembly. Galaxy also has extensive documentation and tutorials, some of which were written by us for assembly. And these workflows, as they're implemented in Galaxy, are super portable, which helps make analyses more scalable, reproducible, and transparent, which are key features of scaling these projects up to multinational and multi-species scales. I just have a quick example of a simple workflow here. So this is just generating a Merrill database from your input PacBio data. And this whole workflow is behind the scenes. The user never sees it when running the workflow. You can edit it, of course, but you don't have to. This is what we basically ship out. And when you import it, then the user just sees this little panel and they upload their data and run the workflow and get all the genome scope plots. So this is a simple example, but we have a more complex one, such as our HiFiasm one. But again, all this is in the back end, so users don't have to run all these steps one by one, don't have to run all the QC, they don't have to worry about any of that. This will just they put in their input data and they will get their contigs and their Mercury plots and so forth. So on that note, you might be wondering where to actually access these. I've put here a lot of key links. So our workflows are accessible at this GitHub. There's a couple tutorials here, and also our genome arc, which is where we put all of our finished and draft assemblies. But you can also just go to this link tree, which recapitulates these links and is a much more easier to type, perhaps, link. So on that note, I would like to thank everyone who helped make this research possible and all the funding who helped make it possible as well. And also thank you for your time.